Okay, good morning, everybody, and welcome back. Welcome back from your spring break. I hope that you all had a good break. You caught up what you needed to catch up, rested where you needed to rest, etc., etc. And today, oops, where am I? Sorry, but today I'm just going to finish off our section on circulation, and in actual fact it forms a good segue into our next section, which we'll begin today, which is discussion about the immune system. Today, gas circulation, we'll mostly talk about the structure of blood and its different functions. And of course, the blood components are tremendously important in the immune system as well. So it kind of segues neatly into that. Blood is an incredibly important tissue. It is regarded as a tissue. It's obviously a fluid tissue. It is in fact a connective tissue, but it has many different components and many different functions. And these are principally in gas exchange. That's the first thing. The second thing is blood is a transport mechanism. It's a transport mechanism, obviously, for the gases themselves, but also for nutrients, hormones, all sorts of things like that. And then lastly, it is intimately involved in the immune system and in the immune function, as we'll see. So the blood is a liquid medium because all of its components are suspended in a fluid, which when we're talking about blood itself in circulation, we refer to as plasma. That is the clear straw-colored liquid, which would be left if you remove all of the other blood components. You're left with this clear liquid, which we refer to as plasma, when it is in circulation in the blood circulatory system. That's the clear liquid which results from the kinds of filtration mechanisms that we talked about before, when blood is pressured through capillaries. That is referred to as lymph, and for reasons that we'll see when we talk more about the immune system. So when we actually, if we were to take a sample of blood and then spin it down in a centrifuge, so that we take all the solid components and we spin them all to the bottom of the tube, and we look at the composition of the blood, we find that about 45% of the blood, of the volume of blood, is made up of various cellular elements, which we'll describe in a second. The rest is this clear straw-colored liquid. We're not going to discuss its composition in huge detail. I'll just tell you that this liquid is incredibly important. And it contains components which are very active. This is not simply a passive fluid to conduct stuff around, or to dissolve things. It actually has components which are active in our physiology. Most of it, of course, is water, because that's the solvent. But then the serum also contains a lot of ions of various salts. And this is how, in fact, all of our cells are supplied with the ions that they need. But it also creates an osmotically favorable environment for all of our cells and all of our organs. Here are the various ions. And sodium, potassium, calcium, etc., etc. But one of the ions which becomes tremendously important is this here, this last one here, and that is bicarbonate. The bicarbonate ion has various functions in serum. First of all, it is a pH regulator. That's the first thing. But second of all, most of our carbon dioxide is transported in the form of a bicarbonate ion. Most of the carbon dioxide which needs to be taken to the lungs for expulsion is actually carried in the form of a bicarbonate ion. So that's why I say this is of particular importance. The next thing is that the serum contains a large amount of protein. The first is this protein called albumin, and albumin I'm sure we've talked about before. It's the same kind of protein that occurs in egg white, and it's a very, very common protein, very abundant protein in most vertebrates. And it has various functions in the serum. And the most important of, first of all, it is another method, another way in which the pH of the serum is buffered. How the serum is buffered to prevent rapid changes, any rapid changes in pH. And albumin functions in that buffering. It also provides an osmotic buffering as well. The next set of proteins that will become important when we talk about the immune system are the immunoglobulins. Now, that is the immunoglobulin. That is the biochemical name for antibody. Immunoglobulins, part of the immune function. Globulin, that is the protein itself, which we'll only describe when we get to talking about the immune system. But they are an abundant protein in serum. Don't forget, we also transport lipids in the blood, in the serum, in the form of those chylomicrons. This is what these are. Don't worry about the rest. This other name, apolipoprotein, is another name for the chylomicron that transports fats. The next is a protein called fibrinogen, and fibrinogen is the protein which produces the effect of clotting, blood clotting, but it exists in an inactive form in abundant quantities in the blood. It is inactive until it's activated by actual blood cellular elements, which I'll describe in a second. There's one thing, though, which I have to say, many of one group of proteins which has been left off here is a set of proteins called complements. And please write that in, COMPLEMENT, complement. And I don't know why they left it off this list, because it's a very, very important component of the plasma. And it is part of the immune system. And I'll describe its function later, but it's important that you know now that complement is dissolved into the serum. So those are then the serum would also be full of nutrients and all sorts of things that are transported in dissolved form around the body. So the rest of it, that's this clear straw colored, 55% of the volume. 45% of the volume is composed of cellular elements, and these are the various blood cells. And we don't need to worry about the actual volumes here, but the first very, very abundant are the erythrocytes. These are these cells here, the red blood corpuscles, and they are what gives its red color. They are by far the most abundant cell. They're a strange cell. We'll see more of them in a minute. In that, in mammals, not in all vertebrates, but in mammals, the erythrocytes have no nucleus. They're still a functional cell, but they have no nucleus. They have limited synthetic ability. So these here. Then if we were to look carefully, I don't know if you can see it on this, there's a thin white layer there at the very top, small by volume. are the white blood cells, which are also referred to as the leukocytes. So the leukocytes are mostly involved with defense and with immunity. And we'll hear much more about their function as we talk about the immune system, so I won't go into it in detail now. The red blood corpuscles, of course, are primarily responsible for transporting oxygen around the body. They contain hemoglobin, which binds oxygen and transports it. The hemoglobin will also bind a small amount of carbon dioxide, but not very much. Most of our carbon dioxide is transported in the form of this bicarbonate ion. So that's red blood corpuscles, gas transport, white blood corpuscles, immune and defense. The last that we need to talk about are these strange little bodies here called the platelets. And the platelets are also sometimes called the fibroblasts. And they are responsible for the clotting of blood. And I told you that the serum contains this protein called fibrinogen, but it's in an inactive form. It's very important that it remain inactive. And this fibrinogen is activated by these little tiny, you can think of them actually as being like cell fragments. They're like erythrocytes that have carried on dividing and have fragmented. Very small. They have this strange property that they remain inactive as long as they're moving. As long as they're moving in the blood, but as soon as you stop them, they'll begin to release enzymes which cause the blood to clot. So the whole blood circulatory system is designed so that the blood movement is continuous, that stuff does not get caught on the sides of the blood vessels, because if it does, there's a great danger that the platelets will cause clotting. So the leukocytes that we mentioned, the white blood cells, I won't go into this in any great detail at the moment, because we're going to cover it in more detail once we get to the immune system. But there are 5 major kinds of leukocytes, and each group has distinct functions and distinct physiology, and they are primarily responsible for surveying the body, surveying all of the tissues, and checking for various things. First of all, for damage. They respond to the damage of tissues. They respond to the presence of pathogens as well. But very, very importantly, and often forgotten about the immune function of these cells, they also survey for tumor cells. They attempt to identify cells which have transformed into likely tumor-forming cells and kill them. So that is their other important function. They can be transported around the body by the circulatory system, but they are also transported by the lymphatic system, and many of them can also move freely through the tissues. They can leave the blood vessels and move through the tissues. That's how they accomplish many of their functions. So all of our blood corpuscles, all of our blood cells, arise from in the bone marrow, in the red marrow of the bone, and they arise from a stem cell. They arise from all arise from a common ancestor. And this is a stem cell, in a set of stem cells. We'll have many of them in the bone marrow, in the red marrow of the bones. But those stem cells, when they divide, they can have various fates. Those fates are often determined by hormonal influences. The body monitors what it needs in terms of the white and red blood corpuscles, et cetera, et cetera. and it carefully shepherds progeny of the divisions into different fates. And these are the different lines. The first line is called the lymphoid line, and this is a lymphoid stem cell gives rise to various lymphocytes. And the lymphocytes we'll see in a minute. I'll show you an actual picture. They are relatively small cells, and they have a very large nucleus that almost fills the cell. The function of the lymphocytes we will discuss when we talk about the immune system. The lymphocytes, two groups, for example, of lymphocytes, the B and T cells, are responsible for producing antibody and various other specialized functions of the immune system. The other line, so that's the lymphoid line, and that is involved solely with immune function. The other line that can be set up is called the myeloid line, and the myeloid line leads to a very, very diverse group of blood cells. It's the myeloid line that gives rise to the erythrocytes, and then to many of the other leukocytes that we see. So the myeloid line gives rise to erythrocytes, neutrophils, all of these things here that we're going to hear about in a minute. All of these leukocytes, including the platelets, arise from the myeloid line. But it's a fascinating thing that in fact they all arise from a common ancestor, and their fates are determined later on after several divisions. So here's what they would look like if you did a blood smear, and I'll just mention the different kinds of blood corpuscle. Again, I'm going to repeat this when we talk about immunity. We'll see more of their structure and function. Here is it. First of all, the first thing you notice in the background here, these are erythrocytes all over by far the commonest of all of the blood cells, but they have no nucleus. They're sort of a donut shape in a way. The center of the cell is concave with a thickened rim around it. This here is a lymphocyte, easy to recognize. It's a smaller cell, and the nucleus almost fills it. When we see a lymphocyte in a blood slide like this, we cannot tell what kind of lymphocyte it is. There are various lymphocytes that we'll hear about. You can't tell. They all look the same. You have to look at their function to determine what they are. This here is called a monocyte. And I tell people that the easiest way to recognize it's a large cell. It has this nucleus with a particular shape. It's like a peach emoji. Can you see? There. So that's the easy way to recognize a monocyte. Monocytes are very interesting because when you see them, this is how you see them in circulation. And you can't tell what their function is when they're in circulation. They have two important functions. They have two important fates, I would say. These monocytes actually enter the tissues of our body. They can leave the blood circulatory system, and they will enter the tissues where they form two. There are two kinds of cells. The first are called the dendritic cells, which we're going to hear a lot about. The other are called the macrophages. And macrophages, their major importance is they are surveillance cells. They are one of the cells that first respond to the presence of pathogens. So a monocyte can form either a macrophage or a dendritic cell. You don't know when you see it in a slide like this. These here are the most abundant of the leukocytes, of the white blood cells. This is a neutrophil here. And you can look at its nucleus, and you can see it has this strange nucleus with lots of lobes to it like this. That's the easy way to recognize them. Here's another one here. They are the commonest white blood cell, and they are the soldiers of the immune system. When the body becomes alerted to the fact that there are pathogens, for instance, if you've got a wound and there are bacteria in it, messages are sent out, and then the neutrophils are summoned to the site of an infection, and they are the cells that actually attack and eat the pathogens and attempt to kill them off. So these are the soldiers, the neutrophils. These cells here we're not going to worry about for the moment. I will mention them again later on. These are cells, eosinophils and basophils, and these are of particular interest. They're a major reason for their existence is to cause inflammation. And we'll see when we talk about immune system, we'll see what the function of inflammation is. These have also become very important in allergic reactions. But we won't worry about them for the moment. Just remember, you've heard the name. The ones that we really want to pay attention to are the lymphocytes, monocytes, neutrophils, erythrocytes, and then lastly, here is a platelet here. You can see how very small they are. They also have no nucleus. They just look really like a little fragment of a cell. And I made a mistake. I told you they were called fibroblasts. They're not. I'm sorry, that was a mistake. The other name for platelets is thrombocytes. I'm sorry, thrombocyte is the other name for platelets. And they are responsible for the set of reactions in the blood serum, which will result in the formation of a blood clot. So this formation of a blood clot is very important. It is extremely complex, a very, very complex set of reactions. We won't discuss it in detail. But when a wound occurs, for example, a cut or whatever, you know that when you get a cut, the cut will bleed for a certain amount of time, then the blood will pool. And when it pools, it begins to form a solid. And that it forms actually like a gel. And that gel does various things. First of all, it plugs up the cut blood vessels and it stops them from bleeding. But it's also the first stage of the healing process. It is through that clot that cells can move to perform the healing of the wound and seal it up again with a scar. So this formation of the blood clot is very, very important. But it's also important in the body that the blood clots do not just simply form spontaneously. That does happen. And when it happens, very often it's extremely dangerous because it can end up plugging up blood vessels which have not necessarily been damaged, and it can end up plugging up essential blood vessels. So, for example, the formation of clots in the coronary arteries is what produces a heart attack. It prevents the flow of oxygen to the blood and blood tissue, the muscle begins to die. A blood clot forming in a vessel going to the brain can cause a stroke. The blood clot prevents blood from flowing to the brain, deprives the brain of oxygen, or a portion of the brain of oxygen. So it's very, very important that the formation of blood clots is carefully controlled, and that it only occurs where it is essential, for instance, where a wound has occurred. The clot itself is referred to as a thrombus. And that's where the platelets get their name from, thrombocytes. They are the cells which are responsible for producing the blood clot. And as I told you, one of the ways in which, principal ways in which platelets are stimulated to cause a blood clot is if you slow them down, if you stop their movement, they begin to interact with the surface that they're on, and they begin to interact with one another, and they begin to release a set of enzymes which will cause the fibrinogen, that protein in the serum, it will cause that fibrinogen to begin to come out of solution and to form threads. It polymerizes to form threads, and the threads actually will trap blood corpuscles in them. And this is one of the examples in our body of positive feedback, because as soon as you start to form this clot, you just have to form a small clot, and you will start to trap blood corpuscles and blood platelets in it. That stimulates the release of more of these enzymes, and it becomes a feed forward. It's a positive feedback. As soon as you get a blood clot, it starts to form more and more and more and more rapidly and more profoundly. So very rapidly, a situation arises where you have this fibrinogen with blood corpuscles, blood cells trapped in it, and that is the thrombus. This is the clot itself. You don't need to, I don't need you to remember all of the details of this. What I do need you to remember is that the principal spur for formation of the clot is blood platelets. It does also, there is also some influence from damaged cells themselves, but it's the blood platelets that are the most important contributing factor. And all I need you to remember is it's a very complex thing. The formation of this blood clot is, if we were to look at all the enzymes and everything involved, you'd see it's a very complex interaction of various enzymes and substrates, which we refer to as a cascade. The details of the cascade you don't need to remember. What you do need to remember is it's complicated for a reason, and that is that it has to be strictly controlled. You do not want blood clots forming spontaneously all over the body. They should only form where it's really necessary. So I said to you that a blood clot can form, for example, in the coronary arteries, in the blood vessels leading to the brain. Why does that happen? Well, the answer is that the blood platelets will start a clotting reaction whenever they are slowed down. So if, for example, we have blood vessels in the heart which have been narrowed by deposition of fat in the walls, for example, blood platelets can be slowed down by that lump of fat on the vessel wall. And as soon as they are slowed down and stopped, they start to produce the enzymes, which can result in the formation of a clot. And that is how a clot can form in a coronary artery. The blood vessels, think back, we talked about the structure of blood vessels. We said the very inside lining is called the endothelium. The endothelium is specially structured so that it is extremely smooth, and all of the blood corpuscles slide over it very easily, including the platelets. They stay in motion because they can't adhere to the endothelium. But if you damage the endothelium, if you lay down fat on it, or you start an inflammatory process there or anything like that, the endothelium can lose function and start to cause clotting. This is a very interesting, and I'll just take a minute to diverge here to tell you. The endothelial cells are one of the cells which the COVID-19 virus attacks. And it's believed that this is why in some instances of COVID infection, there are these strange clotting anomalies. People have problems with abnormal clotting in blood vessels, and it's believed that this can be one of the effects of COVID-19, that it causes the endothelial cells function to change so that clotting is promoted. Here is a scanning electron micrograph of a clot. And you can see here that when these solid threads form, they're called fibrin. Remember, it was fibrinogen when it was in circulation. At the end of the clotting cascade, it forms the solid net of threads called fibrin, and it traps red blood cells, and it traps platelets and stops them from moving, so they release more of their hormones. So the clot grows, and it grows more and more rapidly. Okay, so that's all we're going to talk about for the blood function. We need to talk a little bit about gas exchange and about how gas exchange is affected in the body. All mammals respire, in most vertebrates apart from the amphibians, in fact. We respire exclusively through a lung system. The only vertebrates which have any other system of respiration are the amphibians, which also respire through their skin. All other vertebrates depend entirely upon a lung for their respiration. And all of our gas exchange takes place within that lung system. What the lung actually is an infolding. It's actually what has happened is it's an elaborate tube system with many, many, many divisions. And the reason we have to have this internal lung is that all of our gas exchange has to take place across a moist surface. Our gas exchange, in actual fact, is a system where we move gases from the gaseous form of air, whatever, and put them into solution in a liquid, and then put them into solution, in our case, in red blood corpuscles, which then circulate oxygen, for example. Carbon dioxide returning to the lungs, it returns principally in the form of bicarbonate ion, and is then converted into the gaseous form. And again, that takes place through the medium of a moist surface to put the carbon dioxide back into gaseous form so that it can be expelled. So this is our typical lung system. And in the mammals, one of the characteristics of the mammals is that we have a diaphragm. And it is the diaphragm, this huge muscle that spans, acts as a boundary actually to the thorax. That is a contractile muscle. It's a dome-shaped muscle. And when it contracts, it pulls down, it loses its concave shape, becomes flatter, and increases the volume of the thoracic cavity so that the lungs then fill. In other words, our lungs work almost as by a passive mechanism. We don't actually exert pressure on the lungs to squeeze them flat and pressure on the lungs to open them up by muscles or something like that. Instead, it's a referred system because the diaphragm changes the volume of the thoracic cavity to cause the lungs to expand and contract under atmospheric pressure. So we've done this before, but we'll do it just very briefly again. The opening of the respiratory tract. There are two openings really, the nasal cavity and the mouth. But properly, we should breathe through our nose. And that is because the nose itself has very important functions. First of all, for warming the air. Second of all, for moistening it, for humidifying it. Thirdly, for filtering it. And there is a lot of immune function is dedicated to the nasal cavity because we can take in so many pathogens in through the air. So we'll refer to this again when we talk about immunity. There is one feature of this vertebrate's respiratory tract, which needs to be remembered, is a reference to its evolution. And that is that the upper part of the tract is shared with the digestive system. So the upper part of the tract, the pharynx, is a shared tube. Both food and our respiratory gases pass through the pharynx. We have to keep a very careful check on what is going where. So at the top of the pharynx here, there is a valve system called the epiglottis, which, when we are breathing, shuts off the esophagus. When we swallow, it shuts off the respiratory tract, so that the epiglottis allows food to pass into the esophagus when we're eating, and it protects the respiratory tract from having food pass into the respiratory tract. And when that epiglottis system goes wrong, as it sometimes does, and if any particle of substance gets into the respiratory tract, our immediate response is to start coughing. And coughing will hopefully violently expel anything from the respiratory tract that mistakenly made its way there. The respiratory tract is reinforced with bands of cartilage to keep the tubes open. The place where we want the lungs to be expandable are less well armored with cartilage. But the initial tubes here, called the trachea, are very strongly supported by rings of cartilage so that they stay open no matter what the pressure differential going through them is. The trachea divide into bronchi. A single is called bronchus. There's one bronchus there, one there. Those divide and divide and divide into bronchioles until eventually they divide ultimately into beds of tissue, of almost single layered, cellular layered bags called the alveoli. And here they are here. So this is the ultimate bronchiole is called a terminal bronchiole, and it leads to the alveoli here, looking like a bunch of grapes. Very, very thin walled and very, very richly invested with capillaries, with blood capillaries. That obviously, so that it can be an efficient gas exchange. In the interior of the alveolus is gas filled, not liquid filled. It is gas filled, but it has, of course, a moist lining to it. So I already mentioned the function of the diaphragm. As the diaphragm contracts, it moves down and becomes flatter, and that increases the volume of the thoracic cavity. And in response, to equalize the pressure, the atmospheric pressure pushes into the lungs and expands the lungs. When the diaphragm relaxes, it achieves a dome shape, and that decreases the volume of the thoracic cavity, and air is actually expelled from the lungs. So the lungs have to have a certain, I wouldn't say resistance, they have to have a spring to them. They have to be able to, it has to be able to spring back and forth efficiently. Is there anything else I need to tell you about Yes, the one thing to mention, our respiratory system is not 100% efficient. The vertebrates with the best, most efficient respiratory system are the birds. In the birds, you never get any mixing of oxygenated and deoxygenated air. The airflow is one way. And that is an adaptation in the birds to flight. They have a very, very efficient respiratory and very extensive respiratory system. They don't only have lungs, they have all sorts of accessory organs involved in the respiratory system. We don't. We have a relatively simple respiratory system. And our system mixes, in actual fact, fresh air with deoxygenated air always. We always have a certain amount of residual air left in our lungs. That's just the way it functions, because we just have this one-way flow of air, in, out, in, out. And we always end up mixing carbon dioxide, a small amount of carbon dioxide-rich gas with the incoming oxygen-rich gas. But we're just adapted to it. That's just the way it works with us. So the respiratory system is very carefully monitored, very, very carefully monitored. And we respond to deprivation of oxygen in various ways. We respond by breathing faster, by changing the heart rate and all sorts of things. So there has to be somewhere a very careful monitoring of what is happening with respect to our gases that is in circulation. And this is done by monitoring the blood gas. And principally, the body registers the levels of carbon dioxide in the blood. It's a bit strange. We don't monitor it by looking at the oxygen levels. Instead, the brain stem monitors the levels of carbon dioxide and does it in two ways. It does it directly, but it also monitors the pH of the blood, because the higher the carbon dioxide levels, the more bicarbonate we have in the serum. The more bicarbonate you have, the higher the blood pH. Our normal blood pH is around about 7.4. And so the blood pH, I'm sorry, I said the wrong way around. The blood pH falls with the increase in carbon dioxide. So the normal blood pH is around 7.4. If the brainstem monitors that the blood pH is falling, that is attributes that to the increase of carbon dioxide. This is exactly what happens, for example, if you're exercising very, very rapidly. You begin to build up carbon dioxide levels in the blood. pH begins to fall, and the medulla of the brain registers that. And there are also sensors in the blood vessels and in the blood vessels of the heart, which detect the fall in pH. And that will instruct the respiratory system to begin to attempt to increase the levels of oxygen and decrease the levels of carbon dioxide. The principal way that is done is by increasing respiratory rate, by getting you to breathe faster, to increase the volume of exchange so that you build up the oxygen levels. As that happens, the blood pH begins to rise, and it rises to a homeostatic level, about 7.4, and is maintained there. There's something very interesting about this, and that is that what the body is doing is monitoring blood carbon dioxide. not monitoring oxygen levels. The reason I mention that is because if you become severely deprived of oxygen, if the blood carbon dioxide levels rise very high, there is a set of reactions which we recognize emotionally as panic. Panic is actually a physiological reaction. And in this case, panic arises in an attempt to get the organism to escape the circumstances which are leading to the rise in carbon dioxide. So if you deprive somebody of oxygen for long enough, they will go into panic. And that's a physiological reaction. Why this is so interesting is that we can actually deprive the body of oxygen, but as long as we don't increase the carbon dioxide levels, that panic will not occur. So, for example, if we substitute carbon monoxide for carbon dioxide and oxygen, if we get people into a situation where there's a high level of carbon monoxide, they become deprived of oxygen, but their carbon dioxide levels do not rise. And the body just simply fails to recognize that the oxygen levels are falling until it becomes fatal. But there's no panic, because this is what the body responds to. It's changes in carbon dioxide levels, not changes in oxygen levels. You can, under certain circumstances, you can drop the oxygen levels to fatal levels without the body even really noticing. A similar thing happens, for example, on the rare occasion where an aircraft depressurizes slowly at a great altitude where there's no oxygen, but people in the plane simply fall asleep, go into unconsciousness and die because their carbon dioxide levels don't rise. Their oxygen levels drop, carbon dioxide doesn't rise, they don't register it. Okay, so just to mention that the major way in which we transport oxygen, again, is by binding it to hemoglobin. Hemoglobin is a very complex protein in the erythrocyte. It actually consists of 4 separate molecules which cooperate with one another. Each molecule of hemoglobin actually consists of 4 different hemoglobin components, each of which can bind oxygen. And the reason that happens is because of a particular characteristic of hemoglobin, and that is that when it is in a situation where there is very high oxygen levels, it can bind the oxygen efficiently. But when you take that same oxygen hemoglobin combination, which we refer to as oxyhemoglobin, take that oxyhemoglobin and put it into a situation where oxygen levels are low, it will give up the oxygen. It will liberate the oxygen from the molecule. So as long as there is high oxygen, it binds it efficiently. Take it somewhere where the oxygen levels are low, it releases it efficiently. So in the lungs, hemoglobin very quickly binds oxygen. We then put it into the circulatory system. We take it to the tissues, which are poor in oxygen because they've been metabolizing. When the hemoglobin arrives in those areas where there's a low oxygen level, it releases its oxygen to oxygenate the tissues. It's a very efficient system. The carbon dioxide that is produced by metabolism is mostly transported in dissolved form, in ionic form, in the serum, in the form of a bicarbonate ion. So a single hemoglobin molecule, I told you it has 4 different parts to it. Each of them can transport an oxygen molecule. So one hemoglobin molecule can transport 4, and each erythrocyte is packed with a gazillion hemoglobin molecules. So each erythrocyte can carry a substantial amount of oxygen. And I won't go into detail about it, but these four, in each hemoglobin molecule, the four components interact. If one of them binds oxygen, it makes it much easier for another one to bind. If two of them bind, it makes it very, very easy for the others to bind. There's a cooperation between the hemoglobin units to improve the binding of oxygen. That, I'm afraid, is my hurried rush through the circulatory system. And but it leads us into our discussion about blood components, leads us into discussion about the immune system. The immune system and the immune function, I have to tell you, is extremely complex. And in fact, many aspects of it are poorly understood and only continue to be extensively researched. For us at the moment, of course, any discussion of the immune system is tremendously important because we are dealing with a pandemic at the moment. And the issues of immunity have become daily discussion on the news or wherever. And it really is important for us to be able to understand some of the aspects of immunity, the immune system, and especially the systems which we can manipulate in order to give us protection from disease. And I'm talking, of course, especially about systems of vaccination, which we're all so interested in at the moment. So the immune system is extremely complex. It has several different layers to it. It has several different components, each of which is extremely important. And there is tremendous interaction between the different components to produce a full-blown immune response. But there are some aspects to it which we just don't even really think about as being part of the immune system. So, and you'll understand what I mean in a minute. So these I've already mentioned. These are the three really important aspects of the immune system. It is its responsibility to try and protect us. And in order to do so, it recognizes the presence of pathogens, for example. It can recognize the presence of different kinds of pathogens, bacteria, fungi, viruses. Just for a start, it can also recognize the presence of parasites, worms, that sort of thing. The second thing is it is immune response that recognizes the presence of damaged cells, for example, and especially cells which have become wounded. It is part of our immune system which is responsible not only for ensuring that a wound does not become infected, but also it participates in the healing process of a wound. The third really critically important aspect of the immune system, which we seldom hear about, is that our immune system protects us from potential tumor-forming cells. It is a simple fact that many of our cells divide continuously, and our cells, when they divide, they sometimes divide inefficiently. Especially, we acquire damage to the DNA when in cell division. And this is one way in which, for example, a cell can be transformed. Transformation is the change from a normal cell division, normal processes of cell division, to uncontrolled cell division, which we would recognize as being a tumorous or cancerous cell. And our immune system surveys our body the whole time and eliminates the vast majority of cells which are potentially cancerous. Of course, we know it sometimes misses for various important reasons. But under normal circumstances, our immune system is very, very efficient at eliminating potentially cancerous cells. The way in which the immune system responds to these things, pathogens, damaged cells, tumorous cells, is extremely complex. But in all of animals which possess a jaw, there are two arms to the immune system. The first is a system called the innate immune system. And in the innate immune system is present in all animals. It's present even in not only in animals. It's present in plants as well. Plants also have an innate immune system. So do bacteria, by the way. They also have an innate immune system. And the innate immune system has a set of responses which are pre-programmed. So the innate immune system responds in a single directed fashion to, we could say, an insult, whether it's a pathogen or damage, whatever. However, all animals which possess a jaw also have another kind of immune system, and this is called the adaptive immune system. And the adaptive immune system is adaptive because it can respond in novel ways to the presence of an insult. So the innate immune system does not respond in a novel way. It responds in a pre-programmed way. The adaptive system learns. It actually can see things which it has never seen before, and it can learn to respond to them. And it is that adaptive system, for instance, which produces antibody, which you have probably heard of. So those are two. We'll very briefly discuss the innate system, and then we'll spend some time talking about the adaptive immune system. Okay, so before I finish, are there any questions?
Is there a quiz on Wednesday?
Yes, there is a quiz on Wednesday. Thank you for reminding me. Now, I hadn't forgotten, but I'd forgotten to remind you that there is a quiz on Wednesday. Anyone else? All right then. Okay, so then I will see you on Wednesday. All right. I think because the immune system is actually this discussion of immunity is actually quite complicated. I think I'll make Wednesday a live. No, I can't make a live class because we have a quiz. I will make Friday a live class as well, so that people have an opportunity to ask questions, because this immune system discussion is quite complicated. All right. So I'll see you then.